Hey guys, hello everyone. I hope that all of you are doing great. So, welcome back to the channel. So, I'm going to be starting up with a new series, and this is going to be relating to your the KVS exam, the NVS exam, the EMRS exam. And this video is highly, fully dedicated to the tier 2 part of both the exams. Okay, and here we'll be studying about all the content that you need to know. Okay, and we are also going to do it in the answer writing format because I have not seen a single channel on this entire YouTube which is teaching this. Okay, okay, one thing that I'm going to do is that I'm going to be taking science TGT level here. Okay, so this is going to be the target level here, which I'm going to be helping you guys with. So all of the people who are studying for this particular one are most welcome. Everyone else, if you want to know anything about how to do answer writing and all, you'll be able to get that here. Okay, as well. So yeah. So this is going to be highly focused on this only, and I'm going to be sticking directly to the syllabus, whatever the syllabus we are having, whatever the syllabus that you need. Okay, we are going to be sticking out to that one only, right? So we are going to study all the topics pertaining to biology. We'll be doing biology, we'll be doing chemistry, and we'll be doing physics and biology and environmental. Here both of them. Okay, so we are going to be completing all of the syllabus in within we'll try to complete it within 1.5 months. Okay, let's see how things goes. Okay, because the exam would be around that time only. Okay, so today I will be starting up with this topic of biology, which is cell. Okay, so we'll be studying about cell. Okay, so now let's begin. Okay, so when I talk about cell, so what is cell? Cell is basic building block again, and many of the cells they combine together to form what? All of the cells which have to perform a specific function they together combine to form a tissue. Okay, so say this is a cell, it combines to form a tissue. Further, many tissues they combine to form what? They combine to form organs. Okay, and these organs would further form what? The whole organ system that we are having, right? Like digestive system, etc. Okay, so I just created a rough idea. So here it is going to be forming what? The whole organ system. Okay, now let us start with what is a cell. So basically, what is a cell? A cell is able to show number one, an independent existence. Okay, that is needed. Okay, an independent existence that is needed. Secondly, it should be capable of performing the essential functions of life. Capable of performing essential functions. Okay, fine. So these are two properties that are going to be defining what is a cell. Independent existence as well as capable of performing the functions. So first of all, very firstly, Robert Hooke was involved in, he actually observed dead cells, okay, from a thin slice of cork. We all have known this. These were the dead cells, okay, and he observed that there is a kind of a hexagonal, structures, and they have together formed this cork cell structure. So he basically gave the first idea about what a cell might be, right? Second, who actually put that through was Leeuwenhoek, Anton von Leeuwenhoek. Okay, what he observed, he created his own compound microscope. Okay, and he observed what? He observed those cells, cork cells and other cells under this microscope, and then he gave a precise picture of what a cell could be. A very, not precise, but a very, the most accurate, you can say, the most accurate at that time picture was given by him of a cell. Fine. Okay, so third person who comes into play is going to be Robert again, but this time not Hooke, but this time it is going to be Robert Brown who actually discovered the nucleus of the cell. Okay, so this was a major discovery because once the nucleus is discovered, it would give us the idea about the independent existence part. Okay, so these were the three initial people who observed cell, right? Let's change color. Okay. Now, when I talk about the cell theory, so there is a very, very important concept of cell theory which is always asked, always asked. Okay, it is given by two people. Okay, one is Malthias, I think the spelling is right. Skeleton. I don't know how do we pronounce it, but this is how I pronounce it. Okay, Maldia Skeleton and Theodore Swan. Theodore Swan. So these were the two people, okay, and who gave the cell theory. So what was cell theory about? That Acha, first of all, let us say that this guy, Matthias Schleden, he observed all the plant cells. He actually observed the plant cells and he deduced that all the plant cells are made up of cell. Correct? Okay. Now, this was a zoologist, right? He was a botanist, he was a zoologist. So of course he would observe what? Animal cells. So he observed the animal cells, okay? He observed what? He observed animal cells and he observed the plasma membrane, thin plasma membrane layer that is found in the animal cell, which is found in the plant cell as well, but there is an extra layer that we call as cell wall, which is found in the plant cell, correct? So this was their initial, you can say the initial observations that they made, right? Now, then they came together and they together formulate what we call as the cell theory. Fine. So what does cell theory says? So there are basically three pointers, very, very important pointers. First one being that all the living organisms are composed of cells. All the living organisms are composed of cells and the product of cells. I'm not going to write everything. I'm just saying it out loud so that you guys can write. Secondly, it said that all the cells, all the pre-exist, all the new cells, you can write it, new cells arise from pre-existing cells. That means there needs to be, I have to write this one. I don't know why it just keeps disappearing. So new cells, they arise from the pre-existing cells. This was the second point. And the third point was that all the activities of an organism are the outcome of the sum total of activities and interactions of the cells. That means एक किसी individual में like all the activities of individual is a result of what? The activities in the cell. Activities in the cell. So these three were what? These three were the cell theory part, which is very, very important. Okay, and cell theory part was given by these two people. Okay, and this is very important. This can be asked to you to write it down. Okay, now moving forward, let us see. Okay, so they have in the syllabus they have mentioned that you have to give the like normal idea about this, what is cell and okay. So, if you have to describe cells, so what cells could have? Like if I see a typical cell, if I start from the outer membrane, so there is a cell wall in the case of animal cells, but this is what I made is an animal cell. So like let's write plasma membrane. So outermost covering is going to be of a plasma membrane, okay? And if it is a plant cell, then it is going to be a cell wall also, okay? Then when we move inside, so this all of this area, all this arena where all the organelles are kept and all the activities are happening, this is called as cytoplasm, where all the activities are happening. The central nucleus is going to be there, which consists of what? Which consists of all the genetic material inside the cell, right? And if it is a eukaryotic cell, so you can also observe a nuclear membrane around it. Okay, if it is a eukaryotic cell. If it is not, then you're not going to have a nuclear membrane, and even the that your nucleolus is absent in that case. But if it is a eukaryotic cell, so you can also find nucleolus in it, right? This is the whole this your cytoplasm thing, fine. And what else? Yeah, we have like Golgi apparatus also. Golgi apparatus. We have this Golgi apparatus. We have endoplasmic reticulum, which is rough endoplasmic reticulum and smooth endoplasmic reticulum lying around it. So rough I'll make with some small ribosomes on it. Okay, which is 60S, which we'll discuss. Then okay, then we have a mitochondria as well, right? So a mitochondria can be found in the case of prokaryotic cell. A plasmid can be found. Um, we have lysosomes as well, which are known as suicidal bags of the cell, which are lying around here and there. We can have cell sap as well. Okay, some vacuoles can also be present. Fine. So all in all, if we see, the cell has many different kinds of organelles, which we are going to study now. Okay, one thing I want to tell is that a cell would differ in shape, a cell would differ in size, okay, according to the activities, and there would be different activities as well. For example, if we, why I've highlighted these two is that for the shape part, the shape of the cell could be rigid, as in case of a plant cell, okay, due to the presence of the cell wall, right? And it could be non-rigid as well, as in the case of animal cell, because it just has a plasma membrane on the outer covering, right? As an outer covering. Acha, when I talk about the size part, so very small, it can be a single cell can be very small. जिसका example आता है PPLO, which is pluro pneumonia like organisms. Okay, these are the organism which is considered as a single cell, which is like in a micrometer range, like they are of around I think 3 to 5 micrometer range. Okay, and we also have an ostrich egg as the largest cell. It is a single cell, okay? So it is the largest cell that is known, right? So very small and very large cell, right? So also according to their function, they can be of different kinds, right? Like RBC are of different kind, WBC are of different kinds, then epithelial cells are of different kinds, okay? Then your nerve cells are of different kinds. Okay, so these are all different from each other. They look different, they do different functions. So all of this should be noted, right? Okay, moving on, moving on. Let us come to the biggest major part, first major part, very important. That is going to be about prokaryotic and eukaryotic cell. So prokaryotic, I'll be taking. Please mind my handwriting. I'm just writing on a writing pad and it just gets a bit, it's just a bit difficult to write on it. Prokaryotic cells, okay. Let us study about this thing called as prokaryotic cells. So when I talk about a prokaryotic cell, so of course it is more of a primitive cell as compared to eukaryotic. So all the organelles that would be present in the cell would be of kind of a primitive nature. They would not be fully developed, or they would be in a pre-developed stage, like prematured stage. And there are some other organelles which are special to only prokaryotic cells. So we are going to discuss everything about it. Okay. So first of all, what I want to discuss is going to be the most important distinction between a prokaryote and a eukaryote is through the nuclear membrane. This is the first distinction that anyone is going to be making, that presence of nuclear membrane is not going to be there. Whereas when we talk about the eukaryotes, so the membrane, the plasma membrane would be present. Sorry, not plasma membrane, nuclear membrane would be present. Okay. So this is the point, this is the first initial point that we make if we want to differentiate between them. Also, there are no membrane bound organelles. Okay, so organelles that are found in this type of cells are not membrane bound. Okay, so in the eukaryotes, there are membrane bound organelles like Golgi bodies, like mitochondria, like endoplasmic reticulum. Apart from ribosome, all of them are like bound with a membrane. So this is the second major difference between them. Third difference is going to be that they are going to be having a DNA part, a genetic DNA part would be there. Okay, but they are like not enveloped by the membrane as we have seen. So there is not going to be a particular well-defined nucleus, but rather it is going to be a DNA which is lying in the cell. And plus a very important topic, a very important thing, there would be an extra, not extra, but yeah, another kind of a double standard DNA that is going to be found, which is like not the major one that is going to contain that is going to do all the cell function is this DNA. This is like an extra DNA, okay, that is found, and this DNA is what we call as plasmid. Fine. So there is going to be a plasmid that is going to be found, which is the unique feature of your prokaryotes. Okay, so you can mark this up. This is important. Fine. Actually, this plasmid is used in the biotechnology part, okay, where they use them as a vector in order to introduce the certain genes into the organism. Okay, we'll talk about this in the biotechnology section. Right now, it is not the concern. Now, moving on to the next one. Okay, so let me talk something about the. Okay, let us talk about the membrane, that is plasma membrane, in the case of prokaryotes. Okay, so when I talk about the plasma membrane, okay, so if I talk about the bacterial cells, okay, so these bacterial cells have a specialized wall that we call as glycocalyx. Okay, this is an important one. Okay, so there are three layers in a bacterial cell. First one is going to be glycocalyx, second one is going to be the cell wall, and third one is going to be your plasma membrane. Okay, so there are three layers basically. Fine. So these are the three layers in the case of what? In the case of a bacterial cell. Fine. Some of them also have a layer which is called a slime sheath. Okay, and this is very important in the case of what we call as influenza virus. Influenza virus also have a slime sheath which actually protects it from our immune system. That's why we catch cold all the time. Our immune system cells cannot remember that. Okay, so yes, you can put this here. Fine. Okay, so there is another tough covering that could be found, which is called as capsule. Okay, a tough covering called as capsule can be found. Okay, so these were some layers of the bacterial cell. Okay, and they like this glycocalyx is made up of, you can also write it here, peptidoglycan. Okay, it is made up of peptidoglycan. Right? C-A-N, not O-N. Peptidoglycan. Okay. So, actually, there is a concept of, I have to teach a bit about it. I have to touch it a bit. So, this is about the gram staining. So, what does it says? Gram staining. So, when I talk about gram staining, so gram staining is a staining procedure which helps us differentiate. Okay, which is there are two different kinds of bacteria to be precise. That is gram positive and gram negative bacteria. So in order to differentiate both of them, they have different characteristics. So in order to differentiate both of them, we do the procedure called as gram staining, where we do some like I'll write here, crystal violet is first used. Crystal violet is first used. Okay. Then we wash it with water, and first we do iodine staining. Iodine staining is done. Then there is a complex called as crystal violet iodine complex, CVI complex, crystal violet iodine complex. And then there is a treatment with alcohol, and then there are two possibilities that is there. Some after the treatment with alcohol, like this is a complex, it is going to be stained. The color of the stain is like bluish purplish, so this is stained, right? But when we do it with alcohol, so some of them retain the stain and some of them just don't retain the stain. Fine. So the ones that retain the stain are called as gram positive, and the ones which do not retain the stain are called as gram negative. This is the only thing you need to know, a little bit of information, just so that you guys can be able to know what the concept is if it is asked. Okay. Okay, so we were talking about plasma membrane. So there is a very important concept on plasma membrane that we are going to be talking about, which is related to plasma membrane. This one is related to the structure of plasma membrane, which we are going to take in detail in eukaryotes only. अभी हम सिर्फ prokaryotes की नजर से इसको देख रहे हैं. Okay, only prokaryotes, from the eyes of prokaryotes we are looking at this. Okay, so plasma membrane is a semi-permeable membrane. We all know it does not allow everything to pass through it. It only allows certain things to pass through it. Okay. There are certain extensions of this membrane that is called as mesosomes, which we call as mesosome. Okay, and these mesosomes, they are like in the form of vesicles or tubules or lamellae. So they form like vesicles, tubules, and lamellae. Okay, in the cell. Fine. So just you need to know it here. ज़्यादा नहीं होता है इसमें, ठीक है? We will talk very much through, like the major details would be talked about in the eukaryote parts, because in that part plasma membrane needs to be discussed very elaborately. Okay, we need to discuss the phospholipid bilayer model by Sanger. Here it is only this much is important. You don't need to do anything else. Now let's move forward. You can have a basic idea about your filaments. Okay, the filaments are having three parts. Okay, no, not filament, sorry, I'm so sorry, my bad. Flagella. Okay, the flagella is the one which is helping the cell to provide it mobility, and there is a structure also known as cilia. Like if this is the cell, right? So this long tubular structure which is going to help it gain mobility is going to be a flagella, and the one which is like the short, small structure which are protruding out of the plasma membrane is called as cilia. That is also helping the mobility part of the cell. Okay. So if I'll be taking this, so it is made up of the cilia. The flagella is going to be made up of three different parts. Okay, three different parts. That is going to be filament, the hook, and the basal body. The filament, the hook, and the basal body. Fine. There within there are like ring-like structure within the flagella, and all of this arrangement is going to be different in different cells. Okay. This structure, this particular flagella structure, is made up of a compound called as flagellin. It is made of the compound called as flagellin. Okay. Hmm. Acha, we can also have a concept of pili. Pili is basically a kind of a conjunction tube which helps in the exchange of genetic material. You can also call it as mating. Exchange of genetic material is done by this particular tubule. Okay, pili. We call this pili. It is made up of pillin. You don't need to go too much into it. Okay. Now let me come to the next topic. I think we are done with this. Next topic is ribosome, and we are still into prokaryote. Please mind this. Ribosomes and inclusion bodies, we can say. So ribosomes. Ribosomes are first of all non-membrane bound in both eukaryotes and prokaryotes. Okay, they are non-membrane bound. First thing to be noted. Secondly, they are made up of subunits. They are made up of subunits. So there are two. First of all, there are two types of ribosome. That is 70S and 80S. When I talk about the 70S one, right, which is found in the prokaryotes. So the 70S is made up of two subunits, that is 50S and 30S. Please, if you plus them, so you'll get 80. But the subunits of 70S is 50S and 30S, so don't get confused. Okay, don't put it under the 80 wala part. It is for this only. It is for the 70S. Okay, so S is for Sidberg unit actually, but Sidberg unit, and it is a kind of a measurement. It is a kind of a protein measurement. We need not go into the details of that. Okay, now what do the ribosomes do? Ribosomes can be found on like endoplasmic reticulum, as we've seen, the rough endoplasmic reticulum, and hence this is responsible for the protein synthesis in the cell. Okay, and some of the ribosomes, they would get attached to some mRNA as well. Okay, and they form a polyribosome chain, which we are going to be studying in the DNA structure wala jo part hai. We'll study in that also. Okay, how do this work? Translation, transcription, we have to do. Okay, so we'll do it afterwards. Abhi you don't need to go into detail of this. So this also helps in forming what? A polyribosome, which we also called as polysome chain. Okay, fine. Now, what are the inclusion bodies part? Inclusion bodies bhi aa jata hai. It is written specifically in your syllabus. So when I talk about the inclusion bodies, so these are like the reserve material which are stored in the cytoplasm. So I'll just write it as reserve material, which is stored in the cytoplasm of the cell. Okay, and they lie freely in the cytoplasm. Okay, and they are not bounded by any membrane. I'll give you an example like phosphate. The phosphate granules, in the form of granules, it lies into the cell, right? And glycogen granules, दो लिख सकते हो आप, glycogen granules, okay? These all lie in the cell, not membrane bound, and they are basically the storage units in the form of reserve material it's stored. Okay, fine. So I hope that this much is clear. This was all about your prokaryotes, and we have done the prokaryotes part. I'm going to end this video here. Okay, and we are going to make the next video in which we'll be moving on to eukaryotes. Okay, and we'll be moving on into all the organelles of this. Okay, the next video might be a bit longer. This one I'm going to stop it here only so that the things don't get messy, because the eukaryote part is going to be a good one. Okay, it is going to be a large one. So I hope till here everything is clear. If you have any kind of doubt, the comment section is there. You can write it in the comment section. Keep the comments respectful. Kindly only talk about the exam related and studies related questions only would be answered, and would be it is very like you need to keep the comment section respectful, as it is kind of community where we need to work together. Okay. Okay, so comment section is open. For any doubts, you can put your doubts in there. Okay, I'll answer it in the next video, and the next video is going to be about your eukaryotic cells, and in detail all the organelles would be studied. Okay, so stay tuned and just go like and subscribe to the channel. And I've started up with this series, and this series is going to be a good one, and we are going to complete all the chapters of physics, chemistry, biology. Everything is going to be covered here, and the type and the content depth is going to be so apt that it is going to be the exact depth that is going to be asked in the exam. So it is going to be exactly that. Okay, so you don't need to worry about anything else. So thank you so much. Have a very nice day. Bye.